Join audio, share screen, invite others. Participants look link है. Okay.
Prakash?
Sir.
Any problem?
No, sir. No problem, sir.
Okay. Participants వచ్చారా? ఎంతమంది వచ్చారు?
Present 35 members ఉన్నారు, sir.
Nandu joined us.
అన్న మదన్న
Prakash. Sir, speaker भी बरकत नहीं आ रही. Speaker का join ना लेगा.
ఇంకా రాలేదన్న.
ఏదో మిస్టేక్ జరిగింది ప్రకాష్. ఒకటి నాకేదో ఈమెయిల్ వచ్చింది. ఇదే మీటింగ్ జూమ్ మీటింగ్ క్యాన్సిల్ చేసినట్టు ఈమెయిల్ వచ్చిందంటే
ఐ థింక్ అది 10 o'clock అది
లేదు 11 AM 11 AM షెడ్యూల్ మీటింగ్.
ఐ థింక్ ఇందాక
ప్రెస్ చేశారా ఏంటి?
లేదు సార్. అయితే ఇందాక ఒకసారి క్యాన్సిల్ అయింది కదా సార్, అది ఒక కాల్ వచ్చినట్టుంది. ఇప్పుడే నందకుమార్ సార్కి ఇప్పుడే కాల్ చేశాను సార్, యాడ్ అవుతా అన్నారు.
హలో
హలో
హలో ట్రయల్ చూసుకున్నారా ఇంతకు ముందు ఒకసారి?
చూసాం సార్.
Madhucharan? Madhucharan?
Good morning, sir.
Madhucharan, good morning. What happened?
Sir, nothing, sir.
Seems nobody.
Our question waiting, sir.
Okay, everything is fine with Wi-Fi connection?
Everything is fine, sir.
ఈ సైనింగ్ విత్ గూగుల్ అని వచ్చింది అదే జరుగుతుంది.
Hello, am I audible?
Yeah, Nandu. Welcome, Nandu.
Hi, Ravi. Glad that you are here.
Yeah. You are always with me.
Okay, already 17 and 30 participants.
Start
Hi,
check.
Prakash. Yeah. Yes.
Good morning all. Welcome to Pharma Talk, webinar series 2.0, organized by Dhanvanthari Institute of Pharmaceutical Sciences, Sujat Nagar, Kottagudem. The college was established in the year of 2007 by the management of reputed doctor professionals in Kottagudem and college offering courses like B.Pharm, M.Pharm in pharmaceutical sciences departments. The college established with the aim of impacting excellence in the field of pharmacy. Dr. A. Bhaskar Garu, MD, pediatrician, is the chairman of Dhanvanthari Educational Society. To outbreak and fight against the COVID-19, our college have previously organized Pharma Talk webinars with the theme Formulation Development End-to-End on 21st to 24th May 2020. With the huge success in conducting the end-to-end session and good reviews from students, faculty, and pharmacy professionals all over India. With this strength, we are going to conduct Pharma Talk webinar series 2.0 with the theme Scientific Discussion on Pharmaceutical Technology and interactive discussion for undergraduates. The first day session starts with our honorable speaker, Dr. Nanda Kumar, professor in MGR University, Chennai, with 12 years teaching experience in the field of pharmacy. Today, he came forward to explain pharmaceutical technology and interactive question and answers to outbreak COVID-19 in this lockdown period. The Dhanvanthari Institute of Pharmaceutical Sciences cordially invites Dr. Nanda Kumar sir to continue his lecture.
Thank you. May I switch on my video and my presentation? Few days.
Okay, sir. Carry on, sir.
Hopefully, my screen is visible.
Yes, sir.
Fine. So, stop my video so that there won't be any distractions. Okay. Very good morning to all the participants who have gathered through the Zoom. At the outset, I would like to record my thankfulness towards Dhanvanthari Institute of Pharmaceutical Sciences for giving me this opportunity. When I was approached to join an interactive session with the undergraduates, I immediately liked the idea and gave the consent to Dr. Ravikant that I would join this discussion. Because I believe in this word of, Michelangelo, ancora impero, which means I'm still learning. Michelangelo said these words at the age of 87, and you all know that Michelangelo is such a famous Renaissance painter and sculptor, and his works are still standing very high in the world. Even the Basilica of Rome is designed by him, and he said at the age of 87 that I'm still learning. If Michelangelo says that words, then how about us? We all should be learning. Throughout our lives. So I thought. Today, I should clarify everybody who have gathered that I'm not a speaker as such today, but I have come as a learner myself with the hope to learn few sparkling discussions with you all. But to keep up with the tradition, I thought I would be presenting a talk for some time on some important facets of pharmaceutical technology before we open up the discussion. I also welcome, thank all the organizers for this opportunity. I thank Dr. Ravikant, who introduced me, and I also welcome Dr. Ravikant to join the panel discussions when questions are raised. Because I would like to use his expertise on few areas. So, with all your permission and with all your consent, I would like to, start my, presentation now.
Yes, please go ahead, Nandu.
Thank you. I would like to start from the beginning. We all know that the mankind, as was evolving, diseases were also evolving along with the mankind. So as diseases prevailed, men tried to find out some solution to combat these diseases. And as an outcome, they used all the resources that they had in their vicinity and tried to cure these diseases or to come out of these diseases. And all these happened from the Stone Age, from the shamans of the Stone Age. This practice continued through a number of civilizations. You name it: Egyptian, Greek, Roman, the Indian civilization, the Chinese civilization, Babylonian, Mesopotamian. So all these civilizations, slowly, try to learn the reasons, the background behind the diseases, and accordingly they try to treat them all with the help of herbs and medicines that were available with them. Without, much, knowledge with the pathophysiology, they were able to do that. They were able to cure some of the diseases as per the literature. So where are we now today? We are far ahead today. So today, our understanding on the pathophysiology of disease has reached to a great depth than ever. To the advancements in, these are all because of the advancements in the diagnostic methods, and we are now capable of combating, a lot of life-threatening diseases. Okay, barring, COVID-19, which we are, trying to combat now. Probably we all will come out very successfully even in that. So mankind started, treating diseases by trial and error method, by empirical methods. Later on, because of the advent of technology and the knowledge, we are now capable of treating diseases by working at the gene level. So, it is all because of the, technology, we should say. Now we are capable of doing a lot of things, with the help of technology. So, in the near future, we will be expecting this gene-based therapies coming up. It is all there in the primitive stages, but soon we can, expect in the clinical practice also. So, what I would like to put in my, presentation now is this drug development process. If this particular control is affecting you, let me put it somewhere. Okay, so I'm just going to talk about the drug development process. This is something probably everybody knows very well, because most of you who have joined this meeting are, I mean, UG students or PG students. So you should have heard this, I mean, drug development process at one stage or the other of your, I mean, studies. So this drug development process, I mean, comprises of many stages. It is a very complex process involving a huge number of scientists, chemists, and I mean, pharmaceuticians. So they all, I mean, work together to bring out one particular drug or a molecule, I mean, in its dosage form, I mean, to the market. So it's a very complex process and takes a huge time. I mean, starting with the basic research that happens for a minimum period of 2 to 3 years, then goes on to non-clinical studies using animals. And once you determine a little, I mean, efficacy and the safety profile, then we apply and get the, I mean, permission for the clinical studies, which again is divided into phase 1, phase 2, and phase 3. I mean, wherein in every phase, the number of, I mean, participants or the volunteers who take part in the, I mean, clinical trials increase. And after all these phase 3 are done, I mean, we go for new drug application process. Once the permission is granted, if FDA or the other authorities are satisfied with our data, and I mean, we will be given permission for launch of these, I mean, launch of our, I mean, medication. And once it comes into the market, again, we don't stop our, I mean, surveillance on the, I mean, medication immediately. We keep following it up, I mean, for a long time, and that comes as the, I mean, phase 4. Okay, so this takes a long time. I mean, not small. It is nearly 15 to 20 years. Probably here in this, I mean, image, you would be able to see that, you know, I mean, when we actually start with nearly 10,000 molecules. I mean, thanks to the robotics and the technology that we have, we are able to screen so many number of, I mean, molecules at a time. And even in spite of that, I mean, if we are starting with 10,000 molecules, we actually end up only 250 molecules that actually goes to the clinical trial and non-clinical development, and only 5, I mean, molecules as such reach the clinical level. And out of this five molecules that enter the clinical trial, I mean, hardly one comes. Sometimes even that doesn't come also. So our country is not in a state where, you know, we will be able to invest this much of time and this much of, I mean, money to put into this R&D development. And probably that is the reason where our R&D is, I mean, focusing on only dosage forms rather than the development of a new drug molecule. Whereas all the developed nations who are able to spend a lot of, I mean, funds over R&D were able to, I mean, get through all these molecules. So, I mean, basic understanding of this, I mean, particular, I mean, drug development process is very important. I mean, why I have taken up this in this discussion is because, okay, now we are not able to, I mean, manage all this huge sum of money. I mean, I'll tell you the exact figure later. It actually takes nearly $40 billion at this level. So, I mean, a huge amount of money as well as time is required for the development of medications. So, don't we have an alternate for this? That will be the question that comes into most of our minds. And I mean, there is a way to come out of that. And most probably, I mean, this is the solution for, I mean, combating this problem. We can either, I mean, yeah, there are old drugs available. I mean, why should we go with a new molecule? We are searching for a better molecule with better properties. So if you are looking for better properties, why go for a new drug? Why not change the properties of the, I mean, existing drug? That is the question. So, I mean, you have a person and you just don't like his clothes. So, I mean, you don't change the person. We simply change his clothes, I mean, to a better cloth. So that is what we are, I mean, using here. I mean, old drugs and new cloths. So instead of, you know, using the conventional dosage forms, if we are going with new dosage forms that can overcome the pharmacokinetic problems of that drug, then certainly we will be able to, I mean, alter the pharmacokinetic properties of the drug, and thereby we will be able to, overcome. I mean, we will get a better therapeutic benefit of the same old drug instead of going for a new drug. So the first strategy that we can adapt, I mean, instead of going for a new drug molecule development, is developing new dosage forms for the existing. The second, strategy that can be adapted, is you can repurpose the existing, drug for a new indication. So that is what we are trying to do even in this COVID situation. So we don't have, any drugs, especially for, COVID-19. So we are looking for drugs that are already present, and we are trying to see whether we can repurpose this particular drug or a vaccine for, use of, treatment in, COVID-19. So that is how my anti-malarial drug has come into play. And that is how, your already existing antiviral drugs have come into play in the treatment of COVID-19. So we are trying to repurpose the existing drugs for new indications. There is a classical example. Aspirin, we are known as an analgesic and anti-inflammatory, drug, an SAID. But because of the large dose, because of the side effects that are associated with aspirin, now, aspirin has been repurposed, for antiplatelet. We have been using this since decades. So aspirin is used as an antiplatelet agent. So that's a repurposing. Coenzyme Q10. It is one of the drugs, a supplement that is used in neurological conditions by the neurologist in most of the times. But later on, it was, then used for cardiovascular diseases. So we are repurposing. We are finding out whether the existing drug may have some other effect. And if it has got that potential, why not use it for a new purpose? So that is the second strategy. So, either, develop a new dosage form for the existing drug or repurpose the existing drug for a new indication. These two strategies can help us out. From a huge sum of, comparatively, development of a new drug molecule, this will be, costing you less. And this will also, help you to save a lot of energy of your, workforce. So, with that, I just move on to my, next part of my discussion. Okay, which is here. I'm just trying to, see. Yeah, now it is coming full. So I'm just going to talk, about the conventional dosage forms that we have. I'm slowly going to a different platform later on. So I'm just going to speak on, what is this conventional dosage forms are. Hopefully, everybody in the meeting would be knowing about the conventional dosage form. So I'm not going to talk, in detail about their manufacture, their formulation, and other aspects. Simply, I'm just going to speak on, what new things have come in these conventional dosage forms. Probably that will be the, most apt discussion that we could have. We know, but I would like to emphasize that tablets still today, with the advent of as so many number of dosage forms available, tablets are still the most preferred dosage forms because of their ability, to deliver with more precision. It is a unit dosage form. It has got a very, accurate amount of dose, and it is much suitable for a very large scale, production. It's, it has got a huge market. So any, manufacturer would like to manufacture this drug in the form of a tablet so that it will cover a large market space. Okay, but we have so many number of, different types of tablets, since a long time. And I have just, tried to show you up on few. We have the standard compressed tablets, we have the multiple compressed tablets, we have the sugar-coated, film-coated, chewable tablets, sublingual tablets, buccal tablets, vaginal tablets, dispersible tablets, soluble tablets. It is a huge list, and it goes on. And these are the conventional tablets that we have been using, since a long time. But yet we have some troubles while taking these conventional dosage form. We might not get the, complete bioavailability most of the time, since it is, orally absorbed because of the formulation aspects. We may not get 100% of drug that is going into a systemic circulation. So there is always research going on in the area of, improvising the tablet dosage forms. So as a result of these improvisations, as a result of the innovations that have taken place in the, conventional dosage form area, a large number of or different types of tablets have evolved, which can overcome many troubles that are encountered in, tablet dosage forms. So, I have listed here as a table. I just don't want to, go into, discuss about every one of these. So I will just simply give you an overview of it, and then I will just move on to, discuss about them. See, I mean, oral extended release tablets. I mean, enteric coated tablets is something that we know very well, and those are categorized as delayed release tablets. Okay, and these are oral extended release tablets are nothing but sustained release tablets, which are capable of releasing the drug over a longer period of time. So, I mean, we most of the times, I mean, youngsters in the pharma field might not be very sure. Probably my learned colleagues might, have a better knowledge on this. That many of the dosage forms, tablet dosage forms that we are taking are already, coming under the novel drug delivery category, and they are not, conventional anymore. They are all, novel preparations. Best example, you can see very commonly in the market SR formulations. Many drugs come as SR formulations, and it has become a very common thing happening now. So, these oral extended release tablets can be either dissolution controlled type or a diffusion controlled type. Okay, or it could be even erosion controlled type. So, number of brands have come. I have just listed out few of the brands that are available in the market, like Toprol or Ultramer or Pran. So, all these are, very commonly used. And by Alza Corporation and Pfizer, they have, Alza Corporation's, is a very, a popular dosage form. This osmotic pressure controlled tablets. See, these tablets work very simply on the principle of creating osmotic pressure using an osmogen, and the drug is eluted through a semi-permeable membrane. The drug comes through a small orifice that is made using a laser, beam or light. Okay, so this osmotic pressure controlled tablets are also very commonly available nowadays, and Minipress, Efidac are all, these are, some of the osmotic pressure controlled tablets that are already available in the market. So these are, available over a decade. And talking about, a few novelty in the tablet dosage forms, we have the, probably students in the PG levels might, might have done them, personally. The gastroretentive buoyant drug delivery systems. In simple terms, we generally call them as the floating tablets. So floating tablets can be either effervescent type or a non-effervescent type, which entrap the gas inside, or use the technology of a hydrogel, reduce their density. As a result, they float on the surface of the gastric content. So the ultimate aim of making a tablet float on the surface of your stomach contents or GI contents is just to keep the dosage form longer in your body, so that it will be able to release the entire drug over a longer period of time. You don't want to develop a floating delivery which will release over a period of 12 hours, but your dosage form is washed away within 3 hours. Then there is no fun in giving the floating tablets as such. So you want your tablet to float, so that it will release the entire quantity of drug that is present in it. So that is one technology that has already come into the market. And Medoper is a product. It's a combination of levodopa and benzoxide of Nicholas. And Valrelease is also another. Diazepam is also used. Which is already available in the market. These are all marketed products. So if you are wondering if all these novel things have come into the market, yes, it had already come into the market. The other one is another technology that is used for gastro retention, mucoadhesion, where we use the polymers to adhere to your mucous membrane of your GI tract. They make bondage. These polymers make a bondage with your mucin, and they get connected for a longer duration, stay there, stick to the mucous membrane, and release the drug for a longer duration of time. So all these are the changing trends that is happening in the tablet level. So similarly, the other, even in capsules, a number of technological innovations have already come. We are now no longer stuck only to the conventional gelatin capsules. A number of polymers have come into the market, wherein you can tailor make the release of these capsules. You can even fill liquid into them. So in hard gelatin capsules, even tablet coating technology has evolved a lot. Previously, we used to use a lot of solvent, organic solvent based polymers, which are dissolved, which are soluble only in the organic solvents. Now, hydrophilic polymers have come into the market, wherein ready-made coating solutions can be made, and you can use them for your tablet coating without any environmental issues. So, I mean, a lot of technology changes are happening in the unit dosage form levels. Similarly, we can even find, a number of changes happening in the parenteral dosage forms. In parenteral dosage forms, yeah, very commonly we use ophthalmics, we use irrigation, preparations, injections. So all these are commonly used as sterile preparations, of which injections have taken. We are trying to innovate more, advancements in the injections in order to, reduce down the fluctuations that might happen during a steady state levels. Or we are even trying to increase the patient convenience and compliance, which are still a problem with many of the life-saving drugs. So we are also trying to control the release of the drugs through injections itself. So, for more than one reason, we are trying to innovate even the parenteral dosage forms. And in parenteral dosage forms, while we talk about the, innovations, there are two types of innovations that are possible. One is the delivery device that is, that are going to deliver your, drug. The other one is the formulation itself. So when we are talking about the delivery device, a lot of new devices have come, like, osmotic pumps have come, vapor pressure based pumps have come. What you are seeing is a needle-free, in the image that you see is a needle-free injection. Which will use energy to purge into the skin of the patient. And sometimes the formulation-based innovations are like solid implants. The next slide will comprise of a number of formulations. Which are generally given in the form of an injection. So all these are novel delivery systems, like colloidal dispersions, microspheres in the form of an injection, microcapsules. But of course, microspheres and microcapsules cannot be given by IV, but it can be used for a localized application. Nanoparticles, neosomes, liposomes, resealed erythrocytes, polymeric micelles, and whatnot. All these are the ones that we are going to discuss in the later part of our discussion. And the other important conventional dosage form is the transdermal drug delivery system. Everybody knows, transdermal drug delivery systems are nothing but patches, which are capable of delivering a therapeutic, drug across the patient's skin, and it goes and produces a systemic effect. And these TDDS, the transdermal drug delivery systems, generally comprise of some important components. We all know that, drugs, drug is a major component, but we have a lot of restrictions in using a drug. We cannot use a high molecular weight drug. We cannot use a larger dose of a drug. So all these constraints are there with transdermal drug delivery systems. But yet, in some conditions, it has been very, very effective, especially in releasing this nicotine or nitroglycerin patches have very commonly been used. And apart from the drug, we have the liners, which will protect the patch during storage. We will have the adhesive layer. We have the permeation enhancers sometimes can be added. And we also have the backing layer, which forms the outer environment. So these transdermal patches are known to us. But what is actually happening in the transdermal patch advancements? So with regard to the transdermal patches, a lot of advancements are happening. We can make our transdermal patches in a more sophisticated manner. Okay, whatever I have put on my slide are, four important innovations or advancements that is being done in the, excuse me, in the transdermal system. The first one is called a rate pre-programmed drug delivery system. So this is more of a formulation aspect, okay, wherein we try to, design a dosage form, okay, which will release the drug in a very controlled fashion. So it could be a polymer-based membrane permeation control drug delivery system, or it could be a polymer matrix diffusion control system, or it could be even a micro reservoir controlled system. So by changing the formulation, we are actually controlling the release of the drug from the transdermal patch. That is one of the advancements. Excuse me. Sorry for that hiccup. And now, the other, important advancement that we are making is the activation modulation. How we are going to deliver the drug from the transdermal patches. Revolutionized the transdermal, patches itself. We can activate the, transdermal systems by using physical means. Excuse me. So either by, using an osmotic pressure, or you can use hydrodynamic pressure controlled systems. We can use vapor pressure, mechanical, electrical, ultrasound, whatnot. So all these physical means can be used to activate the transdermal systems. So based upon the activation and the activation strength, the drug will be released into your systemic circulation. That's one way. Or you can even, activate the release from the transdermal systems by means of chemical means also. By, using some pH activated systems or by ion activation, you can do that also. The third one is a very novel thing, feedback regulated drug delivery systems, wherein these transdermal patches release the drug based upon the biochemical changes that is happening in our blood. Say, for example, if a person, has got blood sugar levels, increased in the blood, the transdermal system will respond in such a way that based upon the blood sugar levels, the antidiabetic drug will be released into the blood, systemic circulation. So such kind of feedback regulated systems are also coming up. And finally, carrier-based drug delivery system, wherein we can use some carriers like microparticles can also be embedded in the transdermal patches and can be given. Okay, so this is my first part of my presentation, wherein I have just tried to cover up the overview of the advancements that is taking place in the conventional dosage forms. Okay, if these advancements are taking place in the conventional dosage forms, then are we all satisfied? So that's the question that comes next. Okay, let us take an example of a life-threatening disease like cancer. If the conventional dosage forms that are available are capable of combating the cancer treatment or cancer chemotherapy very effectively, if that question is asked, again we have a vacuum. Yeah, a lot of chemotherapeutic agents are used in the form of a conventional dosage form, but they have their own limitations. A lot of side effects do happen because the drugs are given into the systemic circulation and the drugs are not selective in nature. And again, if you have to, make it very selective, then we have to go for new drug entity, or you have to modify the drug molecule itself, and you have to go by all the processes of the drug development again. So that is again a big trouble. So so what could be the possibility? A man dreamed of this a long time ago, before a century. Paul Eldridge, probably every one of us had heard about Paul Eldridge and his concept of magic bullet. A magic bullet, what is this magic bullet? He dreamed of this magic bullet, which is capable of temporal release as well as spatial placement. These are the two important properties of a magic bullet according to Paul Ehrlich. So what is this temporal release? Yeah, temporal release is a condition, is a property of a dosage form which will make it release the drug in a controlled fashion based upon the requirements. So that's what we call as temporal release. So controlled release with respect to time. And what is the spatial placement? This spatial placement is nothing but specificity. This particular dosage form goes to the area where the drug is required. So specificity or spatial placement and temporal release are the two properties of your dosage form which will make it fulfill the conditions of a magic bullet. But unfortunately, till date, we don't have a dosage form which will exactly fulfill the needs of a magic bullet, but we are getting very closer to it. And and this closing of the dream, closely reaching the dream was possible because of one technology that is happening, and that is our nanotechnology. And in the next part of my presentation, probably I'm going to discuss only about nanotechnology in an outline manner, because it's a very big area to discuss. So we will be looking into only the overview of it. Just for confirmation, is everything going smooth? Dr. Ravi, am I okay to continue?
Yeah, Nandu. Yes, please.
Fine. Okay. So, as I was saying, nanotechnology could be the answer for most of our lacuna in the delivery systems. Nanotechnology, as we all think it's new, it is not very new. It's at least 33 decades old, and it's a multidisciplinary field. It doesn't only cover medicine or engineering; it covers almost every field in the globe. Wherein this nanotechnology deals with, either design, manipulation, you characterize, you produce and apply these structures. I mean, these devices and systems are generally, I mean, what is very, I mean, important here is its size. The size does matter here. I mean, if you have a device of nearly, I mean, 1 to 500 nanometer size range, we call it as a nanotechnological device. And this size is what is going to do the, I mean, magic for us. Let me give a very simple example. I mean, we all, I mean, love the, I mean, yellow
Nandu,
metal. Yes.
Nandu, yeah,
Yes.
with your permission, a small
Yes.
announcement. Third years and final years, please pay attention more from hereafter, because it is specifically designed in your syllabus point of view. So I am requesting all the students to pay more attention now on this. Thank you, Nandu. Please go ahead.
Yeah, with pleasure. So let me tell you this. We all, I mean, as I was saying, I mean, we all love the, I mean, yellow metal, gold. Of course, the price is, I mean, hiking up like anything. This, I mean, we all know that, you know, at a macro level, I mean, when we look at, I mean, the gold, it is in a glittering yellow color. Suppose if you size reduce the same, I mean, gold molecule, I mean, to a nano size, it exhibits a red color. So nano size or reducing a material to a nano size produces uniqueness. I mean, a same material in a nano range will give out different properties, I mean, which is unexpected of. So, I mean, this nano materials generally have a unique and they have a different physicochemical properties than the, I mean, macro compound itself. And that's why nanotechnology has revolutionized all the fields. So this slide probably will let you know, I mean, what this, I mean, it's about the size of your nano devices or nanotechnology. Your tennis ball, I mean, look into this tennis ball and we are talking about a virus. So these are the extremes that we are going to talk about. Your bell pin, or I mean, the pin that we use for starting your papers, the head of that pin is somewhere around, you know, I mean, 10,000, I mean, it is almost, you know, 1,000 microns or almost, I mean, 10 lakh nanometers. And I mean, this example could be, I mean, much suitable one. Our hair follicle has got a diameter of about, you know, I mean, 150,000 nanometers. Think about, I'm talking not about the length, I'm talking about the diameter. So you take your, I mean, hair and cut into, I mean, 150,000 times, and you get one part, and that is 1 nanometer. So it is so small. And most of the viruses, you know, bacteria fall under, I mean, micron size, and most of the viruses fall under, I mean, nanometer scale. And these devices that we are make, they are certainly not going to be 1 micron, but it's going to fall somewhere between 1 to, I mean, somewhere between 50 to 500 nanometers, or even 10 to 500 nanometers would be an appropriate size. Because, you know, less than 10, I mean, probably we'll discuss about that a little later, but less than 10 doesn't work out in the physiology. And I mean, again, if you increase the size beyond, I mean, 300 or 200, it is not, I mean, going to work in physiology. I will tell you the reasons why that doesn't work out. But for nano devices as such, for any field, we generally, I mean, give a range between 1 to, I mean, 1,000 nanometers. But there will be a lot of difference in the literature. Some people would say 1 to 100, some people would say 1 to 500. That all depends upon the application for which we are going to use those nano, I mean, products. But many a times we can confine it between, I mean, 1 to 500 nanometers can be called as the, I mean, nano, I mean, particles or nano devices. Okay, so I'll just move on to the next slide. Okay. Okay, fine. Okay, people had been talking about this nanotechnology for a longer time. You know, are we actually having any nanoparticle related products in the market? If that is your question, I mean, then the answer is your house is already filled with a lot of, I mean, nanotechnological products. Your mobile phone has got so many number of components that those are all, I mean, nanotechnology based. Your laptops that you are using now have got a number of nanoparticle devices. Your computers, your PCs, I mean, these I mean, tennis balls are made up of nanotechnology. Most of the, I mean, many of the fabrics that come, I mean, textile industry, nanotechnology. I mean, we talk about, you know, non-wrinkling, non-staining textiles. All these are output of nanotechnology only. So nanotechnology has already, I mean, reached all our, I mean, shelves. So there is, I mean, no more talks like, whether this nano research will come into, I mean, actual use or not. It had already taken up the places, I mean, decades ago. And now the next question that comes to our mind is, okay, I mean, it has come into engineering and technology. It has come into most of the devices that we are using in other fields are okay. I mean, how about medicine? How about pharmaceutical industry? I mean, nanotechnology has spread its wings in medicine? If you ask, yes, I mean, it had already done that job. So from the consumer goods to medicine, so everywhere nanotechnology has already, I mean, raised its head and it has already spread its wings very well. Okay, I mean, now coming to very specific question, has nanotechnology transformed our pharmaceutical industry? Has it really changed the, I mean, way we look at the pharmaceutical industry, the way we look into the research, I mean, that is happening in the pharmaceutical industry? That is the question that is, I mean, raising in your minds. Yes, I would like to share, I mean, one of the slides, which is an output of the, I mean, decisions that is, I mean, the deliberations of the conference that is conducted by the International Pharmaceutical Federation. Okay, I mean, FIP we say, I mean, very famously. So the International Pharmaceutical Federation, I mean, according to it, I mean, according to one of the reports, it says there are going to be only two fields, it's going to revolutionize the pharmaceutical industry. There are, I mean, they categorized into two. I mean, one is the driving the research field, the other one is the enabling technologies. So what is going to drive the research field is bioengineering, material science, and nanotechnology. And what technologies are going to influence the pharmaceutical industry? Robotics, miniaturization of monitoring and delivery equipment. When I say this miniaturization of monitoring and delivery equipment, we are talking about nano devices that can cells, that can deliver the drug to genes, that can diagnose at the cellular level. So that is the technology we are looking at. And the FIP believes that these technology, this, I mean, nanotechnology is going to drive the research of the pharmaceutical industry in the forthcoming years to a greater extent. Why I'm adding this greater extent is because already this I mean, kind of, I mean, research is taking place. Products are already in the market, but this is going to, I mean, transform completely in a very short, I mean, years to come. Okay, so I again come to my point that I started with. This drug development process is going to take a long time, 10 to 15 years, and it is going to consume a huge amount of 40 billion. As a matter of fact, before 1980s, and it was only $1 billion. I mean, that was spent for drug development for an entire process. But now it had increased nearly 40 times. Why this had happened is because, well, of course, the technology has developed a lot. So, I mean, robotics are involved in the I mean, development of medications, and also, I mean, computer-aided drug design has come into play. With all these technology developments, we should have expected a reduction in the, I mean, total investment for drug development, but it is not happening. Instead, it is increasing tremendously. It is all because the focus on efficacy and safety, I mean, during 1980s was not as stringent as we follow it today. Today, the, I mean, FDA, I mean, or the governing, I mean, the authorizing agencies are very strict in ensuring the safety profile of the patients to a greater extent, and therefore extensive testing have to be done upon the medications before it comes into the market. And that is why this, I mean, raise in funds are required, as well as this is the reason why many of the molecules fail, I mean, during their journey to the market. Okay, so that's where our nanotechnology comes into play. So if you are not able to develop, I mean, new drug molecules, I mean, why not, I mean, turn our researchers, why not do our research towards developing nanoparticles or nanotechnological devices which are capable of, increasing the therapeutic efficacy of these drugs that are already existing. That's what we were discussed in the beginning. So this should be the, I mean, research approach. I mean, this is the approach that is being used nowadays, wherein the drug companies are trying to use the old drug and put it into newer, I mean, devices. Thereby, their efficacy is increased to a greater extent, and their therapeutic efficacy has increased. I mean, therapeutic efficacy would increase greatly, and their, I mean, side effect profile will come down. So that is what we are looking at. Okay, so now, I mean, we are moving into I mean, after having discussed about the importance of the nanotechnology, now we are moving into how this nanotechnology or nano drug delivery devices are, synthesized, or what are the different types. We are going to see the different types of nanoparticles. Of course, these are all overviews. There is every small topic that we are discussing could be a whole subject to discuss about. That much of research has gone into them. So nanoparticles, as we already discussed, these are submicron particles. They are having a size less than 1000 nanometers. Okay, and though we say 1 to 1000 nanometers, the most expected size of them is somewhere around 10 to 200, I would say, for physiological or for injections type. Okay, but certainly not more than 500. Okay, so this definition of nanoparticles would include both nanospheres as well as nanocapsule. We should understand the difference between the two, where nanospheres could be nothing but a matrix device wherein the drug is interspaced, dispersed throughout the matrix. They are not bound at the center. Okay, whereas nanocapsule are those in which the drug is placed at the center and it is completely surrounded by a polymer shell or a capsule. Okay, whereas as a result of their makeup, their release pattern also changes. Okay, so two different types. This can be even extended. The same is applicable even for microparticles. Just a piece of information where those are matrix type, where the drugs are found here and there in between the matrix. As the matrix either dissolves or erodes, the drug actually is released into the media surrounding them. Okay. So now, what are the advantages of these nano drug delivery systems? So if you'd ask me that, okay, I would say, this nanotechnology or the nano drug delivery devices have got a huge advantages over the conventional drug delivery systems. First of all, you can deliver these nano devices, or the nanoparticles through any type of any route of administration. You can give them as injections, you can give them as IM injections, subcutaneous, or if you want to give it orally, yeah, you can give it by oral route, or you want to give it in the form of a transdermal patch, you can. If you want to make it in the form of a localized injection, yes, you can do that. So for any route of administration, these are feasible, and they are going to have a long shelf life. Of course, this particular stability aspect changes from one formulation to another, while liposomes are less stable, while you can expect the metal nanoparticles to be a little more stable. These are depending upon the nature of the nanoparticles. The stability aspects will vary, but you can bring them to a longer shelf life that is satisfactory for a marketed formulation. And they have a very high carrier capacity, like many drug molecules can be incorporated within these within these nanoparticles. And you can many times you can incorporate hydrophilic drugs, or you can depending upon the polymer that you are using, you can even lower the hydrophobic particles. In some of the devices like liposome, you can even put both the drugs in the same device also. A layer can have a hydrophilic drug, another layer can have a hydrophobic drug. Those are all possible. And very, very importantly, we will have an improved bioavailability. Okay, and you can also have enhanced aqueous solubility also. That this technology is also used in the pharmaceutical industry. And finally, one particular property that cannot be that cannot be fulfilled by your conventional delivery system. You can target your nano devices to the cellular level or even to the genes. So that targeting ability, or according to the magic bullet concept, that spatial placement is most possible with your nano drug delivery system. Okay, so now I would like to talk about the different systems that are available. I'm just trying to make you familiar to the different nano drug delivery systems. So we have a huge number or huge types of nano drug delivery systems, starting from polymeric nanoparticles, liposomes, which are the game changers. I will talk about liposomes a little later. So quantum dots, dendrimers, polymeric micelles, metal nanoparticles, carbon nanotubes, neosomes. So a huge number of nanoparticle devices are available. I have just listed only few of them. So apart from this, we have a lot more also. Okay, I mean, you can have, I mean, filarons, which is again the extension of your carbon nanotubes. You can have a nano, shells. So a lot of nano devices are possible. Let us simply discuss about, the salient features are very important points about, these nano drug delivery devices, because my focus is, what is going to happen to nanoparticles as such, rather than, the types, because each one is a different chapter itself. Probably you'll be studying that in your, syllabus later on. Um, Due to time constraint, I will just cover up only the, majority of the points. Liposomes, they generally vary in size from 50 to 100 nanometers, but even, multivesicular liposomes are also possible. Large vesicular liposomes are also possible. These are all very large in size. So you can divide them into smaller sizes also, but liposomes that are meant for, actual delivery into the physiological system will generally have a size between 50 to 100 nanometers, and they are nothing but phospholipid vesicles. So we know that, our body comprises of a lot of vesicles which are made up of lipids, inspired by those vesicles which easily go in and out of the cells. I mean, these liposomes are developed long back in 1969. Probably the pioneer in the field of liposomal development is Grigorides. And these liposomes are already in the market. If I say, for the students, probably it would be a surprise. In 1980, I believe the first liposomal product came into the market, Doxil. So that Doxil is there in our market for almost nearly, 40, to 45 years now. So, so liposomes are not new. They're all, old technology, but still, has got a lot of research going on there to, improvise them. So they are the first, generation, I would say. So they have capability, to, entrap a large number of different types of drugs. As I already mentioned, two different types of drugs can be loaded into liposomes. And we have modified the liposomes in such a way that they can, circulate in your blood for long. So that is my discussion that's following up. So I'll discuss about that later on. Neosomes is a derivation of liposomes, I would say, wherein neosomes use non-ionic surfactants, while liposomes use the phospholipid vesicles. Your neosomes use non-ionic surfactants, or otherwise the properties are going to be much, much similar to them. And polymeric micelles, these are obtained from the surfactants. And by reaching the critical micellar concentration, we will be able to form the micelles. They have a size between 10 to 100 nanometers, but mostly fall around 50 nanometers. They again have the properties of liposomes. They have a very long circulatory capability. We can use them to target. You can fix up some ligands to them, to their edges, and you can target them also. So all these nano drug delivery systems have the capability to both release the drug in a controlled manner, as well as to be very specific to the cells also. Dendrimers are a new entity. Wherein a lot of research is going on the dendrimers. These are highly branched systems, hollow systems, and based upon the number of branches, we can call them as generations. The first, the center part would call as GO, and the next branches would be called as G1. So according to the number of branches, we categorize them. They are capable of, we can, link your drugs to the, the functional groups that are present in their, terminal ends, and we will be able to, load a lot of drug into them. And probably, in the forthcoming webinar, probably Dr. Ravikant will be discussing a lot about it, I hope. And apart from that, we have polymeric nanoparticles also, which are made up of biodegradable polymers. This is one area where we work, where an extensive research had taken place. A number of polymers, biodegradable polymers like PLGA, PCL, or polyacrylic polymers. A number of polymers have been used. For that case, even natural polymers have also been used for making the polymeric nanoparticles. One such is albumin. So albumin can also be used as a material for making these polymeric nanoparticles, and they have proved to be very, very successful. And one great advantage of polymeric nanoparticles is that they can carry a larger load. And you are also have the freedom of combining it with a number of ligands, so that it can be targeted also. So carbon nanotubes is another area which will have very, very smaller devices, 0.5 to 3 nanometer. Think about, they don't even exceed beyond, 10, nanometers. They have, these are nothing but, carbon molecules, and these carbon molecules, they will vary in size from 1 nanometer to 100 nanometer. Sometimes they will have the diameter as 3 nanometers or 4 nanometers, but their length will be even 20 nanometers or up to 100 nanometers are also possible. They can be categorized as either single vault nanotubes, or we call it as multi-vault nanotubes, depending upon the number of layers they have. These, if the one that you see in the image is fileron, which is nothing but carbon 60 molecules of carbon together, form a fileron, a round shaped structure, and you can load drug into them, and we call them as the buckyballs. Okay, and we also have the metal nanoparticles like gold nanoparticles, silver nanoparticles. They have a large surface area because of their very small size. They have a large surface area, and they provide a surface for functionalization. The word functionalization means you are capable of attaching a ligand or a drug for or any other substance you would like to. I'll talk about that a little later. Okay, and so these nanoparticles, metal nanoparticles, can very well be used for delivering the drug, or delivering the drug to a particular cellular component itself. Okay, and quantum dots, again, another area which is coming up very very recently. And these quantum dots are capable of they are more used because of their bright fluorescence nature and their ability to absorb and emit radiations. They are mostly used for diagnostic purposes. Of course, they can be used for drug delivery also, but their usage is more in theranostics. Probably we will have a last word about them later on. Okay, so now I come to so probably I don't say that I have completely covered up everything, but I have just covered up an area to just introduce about the different nanoparticles that are present in the vicinity of a research basket. But now I am going to talk about something which you would be interested to know. So what is actually going to happen? We generally say, we think that okay, we prepare a nanoparticle, immediately inject into our body, and the nanoparticle by magic goes and to the particular place and elicits its function. That doesn't happen. Okay, I have developed a nanoparticle, and this I have tested it outside my body, and I found that this nanoparticle is capable of releasing the drug in a controlled manner for nearly 20 days or 25 days in the form of an implant. Okay, I'm very happy that it is releasing in a very slow fashion. Okay, now I made it in the form of an injection. Okay, of course, there are a lot of constraints to come in, to convert them into an injection. A lot of practical difficulties. There will be a lot of regulatory, constraints on them. Okay, by overcoming all the hurdles, I have made an injection which contains my nanoparticles. Now I'm going to inject it. If I inject it, as such, simply, then what is going to happen is my body is going to remove all these nanoparticles within a span of few seconds to minutes. So after 5 minutes, no nanoparticle is present in our body, but outside my body, this is capable of releasing for 28 days. But inside the body, it is not present even for 5 minutes. So why it is happening? Because this nanoparticle will be considered as a xenobiotic by our body. Our macrophages, we have a system called as the RES, the reticuloendothelial system, which comprises of all these macrophages. These macrophages are nothing but cells that are capable of removing the foreign cells and the foreign proteins by phagocytosis. You would have heard about them, like an amoeboid movement. They can engulf the particle and they kill them. So that's how our body is protected. So when we are injecting a nanoparticle into our body, even those particles will be looked upon by our immune system or by RES system by our macrophages as a foreign material, and it will try to engulf it and destroy it. So that is what is happening. So as a result, once I inject my nanoparticle solution into the body by by injecting into the IV, immediately within 5 minutes, nothing is available. So only if your nanoparticles are circulating in our body for a longer duration, it will be able to find out the site where it has to act, and only then it will elicit its function. But You give them only 5 minutes, then there is no scope of, any therapeutic benefit applied of the nanoparticle. So, this is what will happen in our body. So what we are actually trying to do. Okay, the same technology, yes, macrophages engulf them. So if macrophages engulf them, once it is digested, the drug will be released. So this technology can be used for, diseases of the RES itself. Suppose you have, there is a leprosy, patient, or a leishmaniasis, or there is a particular problem with the, liver. Kumpha cells are present, which are again modified macrophages present in the liver. So they will be, these macrophages will take up the drug and they will, release the drug inside the macrophages, and they can be used for treating the diseases of the macrophages itself. But if a person is having a cancer cell that is present outside the RES system, RES will generally comprise of liver, kidney, spleen, like organs. But if a person has got a tumor in the pancreas, if a person has got a tumor in the muscle region, then how would this technology work out? So it is going to fail. So the most important criteria for us to make the nanoparticle work is you have to make them circulate in the blood for longer duration. If you want them to circulate in the blood for longer duration, then you have to make them escape from the phagocytosis process. So how would I escape the phagocytosis process is my next slide. Okay, so this is what the process that is happening. Okay, let us imagine. Okay, let us consider this diagram. Okay, this was published in one of our papers. So so imagine that this is your nanoparticle. Hopefully you are able to see my cursor. So once the nanoparticle reaches the bloodstream, you know that a number of plasma proteins are present in the blood, and these proteins are abundant in quantity, and immediately they will surround your nanoparticles. I'm just making it very, very simple for you to understand. So yeah, there are a number of factors involved here. So these, to make it very simple, let me say that these proteins immediately surround your nanoparticles and they form a coating over your nanoparticles. So you have sent a device. So that device is completely now covered by these, proteins. These proteins, we call it as opsonins. And this process of covering or, coating your nanoparticle devices by these, proteins is called as, opsonization process. So once this opsonization, why this opsonization is so significant is, so once the opsonization, is done, your macrophages are able to recognize your nanoparticle. Previously, the macrophages doesn't know that a nanoparticle has entered the blood. But once the proteins, cover them and opsonize them, now signals are sent to the, these proteins send signals to the macrophages, okay? And the macrophages now exactly come to the place where the nanoparticles are present. So there are receptors that are exactly, that will fit your, proteins, and as a result, because of the interaction between the receptors of the macrophages and these plasma proteins, endocytosis is, started, and slowly your macrophage will engulf your, your nanoparticle. And the same, the same mechanism is used even in your cells for, releasing the drug. But you don't want that to happen with your macrophages. And because of these cellular processes, finally your, drug is, your nanoparticle is cleared and drug is released because of the, the contents of the cell. Okay, and again, the receptors again relieved and they are back to function again. So this is what is happening. So two processes that will, result in the removal of the drug, removal of the nanoparticles from the blood are opsonization and phagocytosis. So to overcome this, let me, make it a little faster because we are running short of time, I believe. So to overcome this, what can we do about is, we have to look upon the nature. Nature gives the answer for most of our problems. And if you look at, many of the bacteria and many of the, microorganisms use this technique. They are somehow, they are capable of, escaping the phagocytosis. If this phagocytosis is actually happening, even all the bacteria should be removed and we should not be getting any infections. But we do get infections. So by some means, this bacteria are escaping the phagocytosis process. They are escaping the macrophages. They are actually, fooling down our phagocytosis. How they are capable of doing it is because of they are having a hydrophilic layer around their cell wall. That's called a sialic acid. Okay, let us come to an example that we know. Red blood cells. We said that red blood cells are nearly, some micron size. They are colloidal in nature. They are circulating in our, blood for nearly 90 to 120 days. So why not the macrophages are, engulfing these RBCs? Okay, if you say that, they are our own cells, that's why, they are not engulfed. So after 120 days, the same macrophages are engulfing them. So what is happening between the younger RBC cells and the old worn-out RBC cells? So there is some change that is making the, macrophages to engulf them. And that is, because of some, proteins that are present on their surface, as well as oligosaccharides, that are present on their surface. So ultimately, the take-home message is this. So if you want your nanoparticles to circulate in the blood for longer, you want it to have a hydrophilic coating. So most of our, biodegradable polymers, they are all, hydrophobic in nature. And as a result, what happens is they are immediately engulfed. So if I give a hydrophilic coating to them, so they will be able to circulate in the blood for a longer duration. And as a result, they will be able to reach the place where they want to go. Okay, so there are three important factors, that affects this. Escaping the macrophages. The first one is particle size. If the particles are greater than 200 or 250 nanometers, immediately, without any, mercy, your macrophages will engulf them. So you want size somewhere between, 10 to 200. That is the most suitable size for escaping the macrophages. The second one is charge. So many a times your nanoparticles will have charge, and we tend to, have a higher charge because these nanoparticles will remain, very stable if they have a higher charge, whether positive or negative. So with regard to the charges, the positive charge, are generally more prone. They will attract more amount of proteins, plasma proteins, and opsonization process is more for, these positively charged, nanoparticles. Whereas negatively charged nanoparticles and neutrally charged nanoparticles will not attract that much of opsonization process. So they will be able to escape the opsonization process. And thirdly, as I was saying, if you want to escape or if you want to dodge the MPS or the macrophage engulfment, then you should make your particles invisible to them. To make them invisible, coat them with a hydrophilic polymer. And that's where your PEG comes into play. I would like to skip these slides because of the time constraint. So that's where your PEG comes into play. Wherein we just coat them with PEG, polyethylene glycol, which is a hydrophilic polymer. Once the PEG is coated, this is a normal nanoparticle, and these nanoparticles will be engulfed by your, by your macrophages. Whereas once it is coated with this kind of, PEG or polyethylene oxide, polaxamers, even P80 that we are commonly using in our laboratories. So if you are, all these are non-ionic, surfactants, and they are hydrophilic in nature. So if they are coated, upon these, nanoparticles, they will form a shield and they will prevent them from being engulfed. So as a result, what will happen is the proteins will not be able to come and attach here. These, chains of your PEG will prevent them. They will act like a wiper of the car, and they will prevent them from coming and attaching to your nanoparticle. And as a result, there are no opsonization process, or if the opsonization process is happening, that is going to be very limited. And if the opsonization process is not very elaborate, then your macrophages will not recognize them. If they are not recognized, your nanoparticles are going to circulate in the blood for longer duration, and they will be able to, permeate into areas where it has to go. Okay, so polyethylene glycol is one of the commonly used polymer. I don't say that polyethylene glycol is completely free from troubles. It has got its limitations, but it is one of the a most suitable polymer than any other polymers for coating your nanoparticles. So once these are coated, we call them as stealth nanoparticles. If they are, and we call this process as pegylation. Okay, so pegylation or stealth nanoparticles. Stealthing means, being invisible. So once you coat it with PEG, the nanoparticles becomes invisible for opsonization process as well as for the reticuloendothelial cells. Okay, so how much of time am I left with, Ravi? Sorry for inconvenience.
Yeah, nearly 15 minutes, but you can take your time, Nandu, no problem.
It is, see, if it is too long, then, probably, people might feel a little bored. So that's why I'm just trying to. Okay.
Yes,
So
yes, we have 15 minutes left. Yeah.
Okay, I'll just try to wind up within 15 minutes. So, this image that you see is one of, okay, how to, coat this PEG upon the nanoparticle? That will be our next question. PEG can be coated, you can simply absorb them. Physical absorption is also possible. You can covalently, chemically, you can link them to the nanoparticle surface, or, there are other techniques available also. But, I would say, it's very important whether you use physical absorption technique or chemical covalent bonding technique, but you should see through that these PEG molecules are adhering to your nanoparticle till it circulates in your body. So if because your in vivo atmosphere is totally different. So once it is released into your blood, and if all these PEG molecules gets detached, again your nanoparticle is naked and it will be engulfed by your macrophages. We don't want that. So whatever technique we use, we should ensure that these PEG molecules stay with them. Okay, stay with them for a longer duration. And PEG has got another advantage also. We link the PEG molecule, one end of it, to your nanoparticle. The other end can be linked to a particular ligand. Okay, I'll tell you what is ligand in the next few slides. So once ligands are attached, then we can go for actively targeting your nanoparticles to a particular area. Okay, so let me show you. So these are some of our research outputs where we have we measured. These are TEM and SEM images of these nanoparticles. Probably I would like to show you this particular figure. See, PEG is available in various molecular weights. It starts with some 400 molecular weight, and it goes up to 1,36,000 molecular weight. So a wide range of molecular weight are available. It can be branched or it can be unbranched. So it has got a number of types. So what type of PEG can be taken for this is a very important question. A lot of research had been done on that, and we should ensure that a brush-like structure is present upon the nanoparticle, to ensure that they are effectively capable of dodging the opsonization process. And to do that, we generally use a higher molecular weight can be a better choice. And linear polymers, linear PEG molecules are far better suited. So this is the TEM image of our nanoparticle that is developed in our laboratory. You can see a cloud around it, which had formed a brush-like structure around this nanoparticle. Okay, these nanoparticles are somewhere around 200, 180 nanometers. Okay, so this is an enlarged view of it. So you can see all these small dots are a cloud around these nanoparticles. Those are the PEG molecules that have been attached to them. Okay, so so I would like to with the limited time that I have, I would like to talk a little about the targeting approaches that that are present. So of course the targeting approaches are wide range, but I would like to talk about the two important aspects of targeting. There is inverse targeting also, but I would like to talk only about two important targeting approaches: passive targeting and active targeting. Apart from that, we have this combination targeting, dual targeting, inverse targeting. A lot of other targeting techniques are available, but these two are the basics that everybody should be knowing. If you are going to work on the novel drug delivery systems. See, what is this passive targeting? Especially with regard to cancer or inflammatory conditions. So we use this passive targeting. So let me try to explain it very simply. We have our blood vessels, and these blood vessels are made up of endothelial cells, and you can expect these endothelial cells to have a small gap between them. So this through these only we generally this architecture is so well built that I mean, most of our times, these normal nanoparticles will not be able to move through that small gaps. But when it comes to cancer, I mean, cancer cells, they develop their own, I mean, blood vessels, because they need a lot of energy, so they need a lot of nutrients. The channels, the blood channels that are actually supplying them will not be sufficient enough for them to, absorb more amount of nutrition. So at a later stage of cancer, we generally tend to make their own blood vessels, and that process is called as angiogenesis. Since I'm just giving an overview of it, I can explain it to this level only. So angiogenesis is the process of formation of newer blood vessels in the cancer. So these angiogenic, blood vessels do not have the architecture like our normal, blood vessels. They are highly leaky in nature. When I say leaky in nature, they have large gaps between the, individual cells. So these gaps will be somewhere, that can range in rats. It range even up to, 1,400 nanometers. But that is a very rare case. But most times we can expect these, small gaps to be around 400 to 600 nanometers. So there are gaps in the, blood vessels. So these gaps can be used for sending our nanoparticles to go into the tumor, because these blood vessels actually, are a part of your cancer. So if you can reach these blood vessels, so then, our idea of reaching the cancer is reached, because cancer cells do not take, allow your drug so easily. They are, they have a well-developed systems of, pumps, which are capable of pushing all these drugs out. That's what we call it as, resistance, multiple drug resistance. So they push most of the drugs out. So your drug have to be more selective, and that's why we use these devices. So these passive devices can, so now if I have a, a nanoparticle which is covered with PEG, so that, it is not recognized by the macrophages, and very importantly, the size is less than 200 nanoparticles. So sorry, nanometers. So this 200 nanometer sized, stealth nanoparticles can slowly move into the bloodstream. Since it is not recognized or removed by macrophages, it can circulate for a longer duration. And suppose if it reaches the, tumor region, now it can move into the leaky architecture of the, cancer blood vessels. And this process is called as EPR, enhanced permeation and retention. And very importantly, another important point is that there is no proper lymphatic systems that are developed in these blood vessels or in the cancer cells. So as a result, once the cells, once the nanoparticle goes inside, it will not be, pulled out. So it will remain there only. So this is called as EPR effect, enhanced permeation and retention effect, and it is generally considered as a golden standard for, passively targeting your nanoparticles to the, tumor cells. Okay. The other way is the most successful method, is active targeting, wherein cancer cells generally, proliferate a few factors or few receptors more than the other, other cells. Say for example, folic receptors are present in all the cells, but the number of folic receptors that are present in our, in cancer cells will be huge numbers compared to the normal ones. Suppose if you could, attach a folic, receptor, a ligand that can specifically attach to a folic receptor, then, so you have your nanoparticle. Okay, let me take up this diagram probably. Okay, so you have your nanoparticle. Okay, this is your conventional nanoparticle. So this is what we are tailoring it. So we have the nanoparticle, we have the drug molecules inside. So if we can attach this, particular, ligand. So this ligand is very specific for the cancer cells. So they will, so your passive targeting will bring it, through this leaky vasculature, the passively targeted nanoparticle will come inside, but they are not specific to the cells. So they will lay there. So according to the concentration gradient, they will slowly move into the, cancer cells. Whereas here, there is a specific receptor. So there is a specific receptor which will call your nanoparticles. So these ligands are the ones, they are the eyes, wherein the nanoparticle will use these eyes to go and attach with the cancer cells. So this active targeting is the, most reliable method, that is being used in, that is looked upon by the researchers for targeting many of the cancers. So passive targeting and the active targeting are the two important targeting approaches that you should be knowing about. Of course, I have covered only the overview of it. There are a lot of other factors to be considered also. A lot of hydrostatic pressure differences are there within the tumor. Unlike other cells. So those things should also be considered while designing the dosage form. Okay, so coming to the applications, that these nanoparticles can be very well used in cancer chemotherapy. They can be used for, so all these slides talk about the EPR effect. Okay, and they can very well be used for, delivering proteins and peptides, which are otherwise very difficult to being delivered by normal conventional drug delivery systems. Your nanoparticles are capable of protecting them. They can increase their bioavailability. They can protect them against the gastrointestinal degradation, and they will be able to, deliver it to a particular site also. So, nanoparticles can be a very good choice for, delivering your proteins and peptides, of course, very small molecules. And brain drug delivery is also possible. Wherein if you can, specifically attach a particular moiety, your nanoparticles are capable of delivering even to the brain, which is very difficult to, reach by other conventional delivery systems because of the intact blood-brain barrier that you have studied. Okay, and I also would like to introduce to you about theranostics. These are the, talk of the town, I would say. Talk of all the researchers. In the recent, decade, for the past 10 years, we would have got at least, 400, papers in PubMed only about this theranostics. What is this theranostics? We know these nanoparticles can be used for imaging, for diagnostic purposes. We also know that these nanoparticles can also be used for delivering the drug. So one is therapeutic use, the other one is diagnostic function. So if you can combine a single, delivery device, which can both use as a diagnostic, both for diagnostic purpose as well as, therapeutic purpose. Say, for example, a single nanoparticle has got, a diagnostic ability, like, it might be a gold nanoparticle, and it can also have a pharmacologically active drug. So both are, combined together in a single device. Then we call it as a theranostic device, or a, or a nano systems, which have got this, capability to deliver both. Okay, so this is, so you have the therapeutic agents, we have the imaging, agents, both put together in a single device, and those are called as your cancer nanotheranostics. Okay, or for that case, anything. This specifically we have mentioned it for cancer nanotheranostics, but it can be very well used for other, disease profiles also. So now you should be, wondering, are we okay? All these are, for talk, these are all sounds very good. What is the practicability of, getting these nanoparticles to the market or to the clinical application? If that is the question, probably, for all your students, this is the, one slide that I kept to show the important milestones in nanotherapeutics. Like, in 1965 itself, we developed the liposomes, and this doxil, which is a stealth liposome or pegylated liposome, has come into the market in 1995. So all these are developments, we developed these systems, but these market, started in 1995. Myoset, Abraxane, probably, I would specifically ask Dr. Ravikant to discuss about the mechanism of Abraxane on your webinar. On probably we both have discussed about it many a time, but no, I kindly insist you to explain the mechanism of Abraxane to all your students, because that is such an interesting, mechanism to understand, and how these nanoparticles actually work. You would be fascinated to know about it, and probably, Ravi is one person who can very well do that. I believe so. So, and these are all, these are the devices which are already in the phase trials. I have also listed, the number of devices that are there in the market, approved by FDA and other approving agencies. So this is the list of, agents that are already available in the market. Some are used for diagnosis, some are used for therapeutics, some are only, protein conjugates, but all fall under the, nano size, and so we have categorized them in this slide. So again, the list continues. So pegylated interferons, pegylated, alpha 2b. So rapamune is a nanocrystal. So here comes Abraxane, your polymeric nanoformulation of paclitaxel. So these are all, one of the, game changers of the nanoparticle market, Abraxane, I would say. Probably in the next webinar, you'd hear more about it. So I would like to conclude here that, if you have an expertise to work on the nanoparticles in your pharmaceutical R&D, so it has got a wide, application reserved in them. It is going to be a very promising approach for most of the problems that we have in drug delivery systems. Only thing is these regulatory aspects, have to be, have to be solved, and we should design in such a way that we can pass all the regulatory testings, and if we can bring out a safe nano delivery system, it can certainly help a lot of patients to recover from life-threatening diseases like cancer. So I would just say that it is up to us to determine our future. So if your future is nanotechnology, yes, we have a huge scope to solve most of our troubles that we encounter in our pharmaceutical industry and drug-related problems. So I would like to thank everybody for your patient listening, and also for all the organizers. With that, I would conclude my my talk. If there are questions, probably myself and Ravi would be happy to answer. Over to you, Ravi. Over
Yeah,
to
Nandu. Nandu, thank you very much for your nice presentation. Yeah, Prakash, is there anybody ready with the questions?
Yes, sir.
Nandu, do you see some people in the chat box? They are saying that, sir, we are not getting bored. Please ask Nandu sir to continue.
I was in a full screen. I was not able to see. If that was the case, then probably I would have covered a little more aspects of this. Probably in the future, we will certainly be able to do that. Because it goes in a flow. It would have been so good.
Yes. And we can get some time.
I'll stop sharing, and so that I'll be able to see people.
Prakash, anybody? Sir. Yeah, please.
Yes, sir. One first question.
No.
Sir, are there any other examples of cancer drugs except paclitaxel and marketed in the nano practical formulations?
I think, exactly speaking, the first Doxil itself is doxorubicin. The first product itself is doxorubicin. I would say, a lot of drugs are coming up in the form of a nanoparticle, in the sense they are nano size reduced, but successfully coming out in the form of a nanoparticle, polymeric nanoparticle is a question. But of course, in the list I have mentioned, a lot. Probably I'm going to share this slides. So you'll be able to see a number of this particular Marbecue. Here I have posted it here. So this Marbecue in 2012. This is one drug, Vincristine. Probably you should be very much familiar with Vincristine. That's why I've taken up this example. So this Vincristine, is converted to a polymeric form. So that's one, and a lot more also coming up. Many are there in the clinical trials, and I would say, an interesting point here. So a technology that is suitable for one particular cancer drug may not be suitable for all others. So if doxorubicin has been put into stealth liposomes, why not all the drugs in stealth liposomes might come to your mind, but it is not so. Especially with these nanoparticles or micro devices, you have to design such a way that you have to tailor make them for every drug, because the physiochemical properties of each of these drugs are different. I'll tell you best example. As Abraxane is nothing but an albumin conjugated paclitaxel, and people try to, probably Ravi will be agreeing with me that people tried to make a similar kind of a formulation for a lot other drugs, but it all failed. It is because you have to treat them all, differently for different drugs. And that's why, we have a lesser number of, products in the market.
And one more question, sir.
Yes.
Is there any chances of causing diseases or cancer if nanoparticles without drug? I think only gold nanoparticles, copper nanoparticles is there now. Without drug nanoparticles will be entered into the normal cells.
Uh, Actually, that's where R&D actually plays a major role, because during the selection of the materials, it's not only whether it is capable of getting into a nano size. We shouldn't be looking into that. What is the most important factor is it should be a safer, device. So the selection of material is so important, in designing the drug delivery. So if you are selecting a material that is biodegradable under the conditions of your tumor environment, then it will not cause much troubles. But of course, there are a lot of researches had been done on the toxicological properties of the nano devices. Even let me tell you, for the case of discussion, even PET, which is considered to be very safe, is not very successful anymore, because our body has started producing some particular anti-PEG antibodies. So we can't say that whatever we are making are going to be very safe. But whatever has come into the market, till date, had been designed in such a way that they are all biodegradable, and that's why they have come into the market. If you take Abraxane, it's an albumin based, and certainly, it is devoid of any toxicity profile. And the liposomes are considered very safe because of their lipid nature, which are all already indigenous to our body, and that's why they are not very toxic. Again, our body will recognize them also. Sometimes there is a ABC phenomena, probably a little beyond this discussion. So selection of the material is the most crucial while you're while you're fabricating this nano drug delivery system. Ravi, would you like to add something upon this particular point?
No, Nandu, please.
Okay.
Pramod Kumar is right to ask.
Yeah, good afternoon, sir. It was one of the most enlightening this webinar that I heard. And as I said that I was not getting bored, I would like to listen hours together.
Thank you.
It's quite interesting for me when I was listening to your webinar. And the flow was good, that you never get bored. And the topic is interesting.
As a matter of fact, since the webinar was designed for UG students, when I was discussing with Dr. Ravikant, so we made it in a very light manner. So if you're all interested, in the future, maybe a little later we can talk something in more detail. Probably after you study your novel drug delivery system.
You are welcome, Nandu. We will host you.
Yeah. And now everybody is so well, accustomed to this, online systems. So we can have that discussion at any point of time.
Yeah.
So thank you for your comments. Thank you.
Actually, I was working for I was a research scientist in Piramal, where I used to work on nanotechnology and all, where targeted drug delivery system was one of the best system where we used to design our drug. And with the, as you said, ligand based. So ligand based is one of the best approach where you can still target. As you said, there are a lot of folate receptors in your cancer cells.
Yes.
So we can target our drugs, as you said, from methotrexate, and also can be targeted because we have got similar this thing where it can be used for arthritis as well as for cancer and lower dosage. So I think so that this can be a good thing when we work on this. As you said, targeted drug delivery, you can use me as a thing for a targeted drug delivery system.
Yes, sir. Yes. Thanks for your input, sir. Your inputs would add up more information to our students. Probably I've worked on, in an industry from this aspect. There are very few industries which do, work on this area. But I would like to add up one more thing that, though we say active targeting is one of the most promising approaches, we got to be very careful about that because, still, there are debates going on. like whether this active targeting is going to be better because of course it takes your device exactly to the site. But sometimes there are chances that it could even fail because of the loosening of the ligands from the device. These problems do come. So it is a very critical aspect that we have to look upon. Probably, in the research, articles, these things do keep coming. Thanks for your input, sir. Thank you.
Thank you. Thank you.
And one small question, sir.
Yes.
Nanotechnology applicable for probiotics?
Yeah, but we should look into the necessity of it. If probiotics can be, see, nanotechnology is not yeah, a very, I shouldn't be using this normal parliamentary word, but it is not cheap. It is a high-end technology. Probably people who are working in this area in the industry would know. How much of facility have to be infused into the industry to even begin this? At an R&D level, and to produce it is again a big challenge. So, producing with the high-end devices. Because I haven't discussed in my discussion about the methodologies used for nanoparticle production, but different methods have to be used. Can be used, but these devices are very costly, and to bring it to a particular size is a very big challenge. So, high-end equipments are used. You got to lyophilize them. So, for every single stage, there are a lot of devices are required, and at the end, the ultimate cost of this nanoparticle devices are certainly costlier. So, for a probiotic, is this needed? If there are simple technologies available for, giving a probiotic, should we use this nanotechnology? Always research should be, looked upon, from one aspect. Is this research is going to benefit us? More than the existing, product. If that is the case, then we can put in our energy as well as the resources into that research. But if a probiotic is needed too essentially, and certainly nanotechnology has got its scope, to deliver it.
Yeah. Yes, Nandu. To add to your points, as Nandu said, it is not a cheaper technology. And if you see the nano formulations, most of them are for cancer. The need is that we have to target these, cancer APIs so that people are using this nanotechnology. But as far as my knowledge concerned, there is no requirement for the probiotics to be targeted. Yeah, I think that should be answered.
Rather, sorry to interrupt, Ravi. We actually, when we are as a part of our discussions with our students, we even thought about, doing a chocolate-based probiotic, chocolate coating to probiotics. Since don't take that probiotics. So there are simple technologies available. Simply by lyophilization, very well do that. So, that will bring down the cost of a probiotic. You don't want to spend thousands on probiotics, isn't it? Yeah. But for a cancer, for leishmaniasis, for diseases, life-threatening diseases, where you want life-saving drugs, there people can spend such huge amounts on these technologies, but certainly not for probiotics, I believe. But if there is a need, as I told you, and if we can equate it, justify it, then yes, nanotechnology can very well make a probiotic delivery, I believe.
Yeah. Prakash, next.
Sir, and one more question. Nanoparticles, how to clear the body?
Okay, probably if I should understand, they are trying to ask, how these nanoparticles can be cleared from the body, right?
Yes,
that should be the question. Actually, we designed the nanoparticles in such a way that it gets us, that's an important point to be noted, because we are sending some xenobiotic material inside. We are trying to mask them. We are trying to take them into the cells. But after getting into the cells, you don't want that material to stay there for long, because again, that is going to produce toxicity, as one of the persons asked. So we have to consider, materials that are more, fastly biodegradable, as well as, bioabsorbable. Okay, we are talking about reabsorbable polymers recently. where they will break down their chains, and they will be broken down into smaller units that can be absorbed by the body itself, like the biodegradable sutures that have come, which will shed by themselves. Likewise, these nanoparticle, if you are using a polymer, certainly carbon nanotubes and liposomes might not have these troubles. But if you are using polymeric nanoparticles, we have to use a polymer that are reabsorbable polymers, biodegradable and totally biocompatible. So those polymers will not give you this trouble. Of course, they will take their time, but they will be completely removed from the body or absorbed by the body into non-toxic.
Yep. Yep.
And the student's point of view, one small question, sir.
Yes.
What are the characteristics of nanoparticles?
Yeah, that's another. The whole class itself. Probably we can spend a whole class on method of preparations and evaluations. But since it is asked, I'm very much glad to discuss that. See, as I was saying, like any other delivery systems, we should evaluate and characterize these nanoparticles. This is where the regulatory bodies are more stringent about. See, the name nano itself comes because of its size. So evaluating the size and characterizing the size is the most important and the foremost point that we should look at. If the nanoparticles do not get its size, then there is no meaning in testing the other properties at all. So first thing is size determination. For size determination, since they are in nano size, we cannot use any other conventional method that we use for microparticles also. So we generally use the photon correlation spectroscopy. PCS, which is nothing but light diffraction methods. Okay, so this photon correlation technique will be able to measure the size of the nanoparticles at a very faster pace. Probably we'll get the outputs of 10,000 particles will be measured. They are based upon the light scattering abilities of your nanoparticles. The larger the size of the nanoparticles, the more will be the scattering. So based upon measuring the transmitted light, you'll be able to determine what will be the size of your nanoparticles. So photon correlation spectroscopy is the most commonly used method for characterizing the particle size. So of course we can, and similarly, you should study about the morphological characteristics of your nanoparticles also. For morphological characterization, generally we go with scanning electron microscopy. Scanning electron microscopy with regard to nanoparticles, it is highly limited in its use because it will show you only the surface characteristics, whether you will be able to see whether it is clustered or not, but we won't be able to see it completely. We won't get the complete morphological information. So we generally go for higher methods like transmission electron microscopy. This transmission electron microscopy, will give you more idea. You will be able to identify what is the surface nature, whether it is rough, whether it is smooth. As I showed you one of the images, you can see whether pH is coated or not. To that level, you'll be able to see with your TEM. We generally say it as TEM, transmission electron microscopy. And not only that, newer methods have come. Atomic force microscopy has also come, which will give three-dimensional images of your nanoparticles. So these are some devices. Cryoscopic microscopy, it's another one, okay, which can also be used for characterization. Now, we also have another, lot of other techniques like X-ray, X-ray attached, diffraction techniques, which will also give you the functional groups along with the morphological characteristics. Not only that, these are morphological characterization alone. Apart from this, so size, morphology, we have a lot other characterizations to be done. Drug content, and drug encapsulation, properties. Too many, to say, I would say. And release characteristics have to be determined also. We have special techniques for, characterizing the release characteristics of the nanoparticles. Mostly, at research level, we generally use the dialysis techniques, which will give a more reliable data. So, characterization, again, whole class can be spent. I just, listed only, few of the important ones.
Yeah, Nandu, to make a session more interactive, I will add one more point here.
Yes, yes, please.
As you told, these properties which we characterize are different for the different formulations.
Yes,
There are
yes,
some
yes.
common and some common characteristics that should be evaluated. There are unique, like for example, if we take albumin-bound nanoparticles, we see the ratio of drug to albumin that is unique for it. And we see the type of albumin that we take, whether it is a oligomer or polymer or monomer, something like that, how much percentage of it in the albumin. And if we take liposome nanoparticles, so we see the lipid content, cholesterol, and the drug that is encapsulated inside of the core, the drug that is present on the periphery, something like that. So the characteristic parameters are, these parameters are different for different formulations. And also, the companies who are developed continuously gives the feedback or continuously educate the FDA also. It is very interesting to us. We feel that FDA knows everything, but since these nano formulations are very, very complex, very new, even the FDA people need to be educated on that. So these companies who are developed these formulations, time to time, they send the data to the FDA and tell them how this formulation is unique or innovative. So that when the generic formulation come into the market, the FDA check all those parameters, whether it is equal to the innovator or not. So that is how it goes.
That's
okay,
true.
next,
Ravi. I mean,
Prakash.
whatever, as Ravi noted, I mean, for different,
yes.
I mean, we have different types of nanoparticles, and accordingly, I mean, different parameters would change. So whatever I narrated in the beginning, they are all, I mean, general ones for all the nanoparticles, probably particle size and I mean, this SEM and TEM, I mean, scanning electron microscopy, these are all common ones. What Ravi was, I mean, trying to say is, I mean, it's very, very specific. As he said, for liposomes, it is going to be different. And for, I mean, metal nanoparticles, it's going to be different. And very importantly, we also determine the surface charge, isn't it, Ravi? So that's another important parameter that we look upon. And we, apart from the release, we also look into the antigenicity of the, I mean, final, I mean, product. We look into the biocompatibility and toxicity, degree of the biodegradability if you are going to use the polymers. So these are all, I mean, huge things to discuss. Probably as already mentioned in the beginning, a separate, I mean, discussion forum is required for this.
Yeah. Yep. Prakash, anything else?
Oh, okay, sir. Thank you, sir. Madhu, sir.
Yes.
Madhu, sir.
Yes, sir. Yes.
One more question from student.
Yes,
sir. Are there any drugs for assumptions from nanotechnology?
Come again, sir. I didn't hear you clearly.
Are there any drugs for assumptions from nanotechnology?
Assumptions.
Yeah.
I didn't get that question very clearly.
Not clear, actually.
I think already developed drugs from nanotechnology.
Marketed.
Yes, that's what I have shown you the list. I mean, wherein a lot of existing drugs only. I mean, we have tried to develop. Okay, let us take the best example of Abraxane itself. Paclitaxel is a drug, I mean, which has got a lot of troubles with its solubility. I mean, to answer your question, so a lot of troubles with its solubility. Initially, I mean, people are finding it so difficult to give the, paclitaxel, to the patients, because paclitaxel is very effective in, reducing the cancer. Cancer cells have, it acts on a number of, cancer cells during its development itself. And now it is, it's the first choice for breast cancer, cervical cancer. But even today, the conventional delivery system of paclitaxel is given with a lot of difficulties, because paclitaxel itself has got a number of, side effects. Like it produces a lot of hypersensitive, reactions. So what we generally do is, in the clinical practice, people have to be pre-medicate, the patient to reduce the, some sensitive reactions. So some allergic reactions, to combat them, before giving paclitaxel, some medications have to be given, some antihistamines have to be given. and you cannot give paclitaxel in a very simple form. I mean, like simple infusions. There are separate infusion sets available for giving paclitaxel, because paclitaxel is not soluble in water. You have to solubilize in a special kind of a solvent. Okay, it is a mixture of castor oil and ethanol. So we call it as cremophor. So this itself is having some neurotoxic problems and nephrotoxic problems. So to overcome this problem, so this existing drug in its conventional dosage form has got all these disadvantages. To overcome this only, people are trying to develop novel drug delivery systems, nanoparticle drug delivery systems for paclitaxel, one of the most researched drug for this purpose. And once they came out with Abraxane, which is an albumin conjugated paclitaxel, its effectiveness is increased to many folds, and its side effect profile has also come down a lot. So compared to paclitaxel as such conventional, your Abraxane nanoparticle form is far better, and it has increased the market space of paclitaxel to a greater extent.
Sir, one more question.
Okay. Can we
Sir,
bind
one
up
more
this question?
Yes, let us hear it.
Okay. Yeah, please.
Sir, we use medication for cancer by nanotechnology, and can we use it for COVID-19, sir, in the same way?
Okay,
Question by one student.
I was actually expecting a question on COVID-19. I was a little surprised that it was not coming all this time. As per the problem with COVID-19 is, with regard to nanotechnology, what we are trying to do is, as I already mentioned in the beginning, we are trying to put the old drugs into new cloths, new delivery systems. So that is the major important advantage of nano delivery systems. But here, with regard to COVID-19, we don't have the drug itself. Okay, so that's one point. So if at all we have a drug and if we have problems, we can use the nanotechnology. But to tell you, the it has been reported recently in some of the research articles that if you can, the antivirals are, commonly used antivirals are used in SARS condition. So this one of COVID-19 is also a SARS type. So if it can aerosolized in a nanoparticle form, the antiviral activity of these drugs are increased, and it is more effective against COVID-19. One of the research says this. And for answering this question, I would like to put up one more, important point. All the companies that are trying to develop the vaccines for COVID-19, I should say they are all working on nanotechnology only, because virosomes is a type of nanoparticle, transferosome, ethosomes, virosomes. These virosomes are nothing but we use the envelope of the viral particles itself, and into that we are actually giving it as a vaccine. So already vaccines that are present in the market, like Inflexal, which is a influenza virus, itself is a virosome pool only. So such a kind of vaccine can be expected, wherein we use the virosome pools, and which is again a nanoparticle type, or we can use viral particles as we use in the cervical cancer. So there are products, there are vaccines that are based upon nanotechnology that are there in the market already. Probably the COVID-19 vaccine that are going to come up soon, probably they can be virosome based. I expect it so.
Okay, thank you, sir.
Are there any questions, Prakash?
Yes, sir. Madhu sir,
Thank
please
you
vote
so
of
much.
thanks.
Thank you so much, sir, for your valuable speech. From the whole management of Dhanvanthari Institute of Pharmaceutical Sciences, it is very interesting, sir. Your webinar preparation, your webinar presentation is,
Thank you.
and wholeheartedly we visit you once again and once more, sir. We are expecting these kind of webinars.
Yeah. Soon we can arrange for that. Thank you very much for the opportunity, and I thank Ravi for introducing again. It is, even for me, it is a very nice experience to, talk to, students of yours, and also, delegates who have joined the webinar. Thank you very much. I look
Nandu,
forward to talk to you all once
I
again.
have a small request from Dhanvanthari College. Your panelists, actually, Nandu, though have command over vocabulary and have good scientific writing skills, he never tried to master the scientific work. I know Nandu from 2007, he has been same and he has put continued efforts in spite of many difficulties. Nandu, I request you personally to take one class for our faculty members and students on scientific writing. Because,
Okay, Ravi.
yeah, yeah. Yeah, is this possible, Nandu?
We'll try to plan it very soon, Ravi. Because teaching teachers is not as easy as we do it for students. So that too on scientific writing, a lot of inputs have to be infused. We'll do that in a short time. Though within this lockdown period or after, certainly we will do that, because that will help me out also. To recap all my skills on that. So I will work it out, and I will certainly come out with a very good output. Sure, I'll take up that.
Thank you very much, Nandu. Yeah, thank you.
Thank you, Ravi. Thank you very much for arranging this, and thank you. For all the organizers of Dhanvanthari Institute. So thank you very much.
Thank you, sir.
With all your permission, I'll take it.
Yeah, yes, sure, Nandu.